So I just want to thank Jim and Linda for inviting me. And I also want to start off by thanking all the people that have contributed to the research that I'm going to talk about today. So I'm going to talk about 3 different projects that are represented here by a great group of co-authors and collaborators and funding agencies. So plants and other organisms play host to whole communities of microbes. And a really useful way to think about these communities is by grouping them spatially. So in this diagram by Hassani and co-authors, there are communities of plant-associated microbes that are grouped by whether they're in the soil or the air, those that are more close to the plant, the rhizosphere and phyllosphere, and then those that are within the plant. And spatial organization is really important because their interactions can depend on how far away they are from each other. A couple of factors that contribute to these interactions are immune reactions of the plants that they're interacting with and resource overlap between the microbes. So those 2 factors can contribute to these interactions. And I should have said before, each of these dots represents a microbe and the lines are their interactions. And so some examples of these resource and immune-mediated interactions. So immunity can have a negative, it can mediate negative interactions between microbial groups. In this example, arbuscular mycorrhizal fungi and rhizobia increase callose production in wheat, and then that has a negative effect on pathogens. And so we have this sort of, it's cross-protection is what it's often referred to. On the other end of the spectrum, we can have positive interactions through plant immunity. In this example, one microbe allows a plant that is normally not a host to Phytophthora disease to become infected. And so that's something that's often referred to as immunosuppression. Resources, on the other hand, can also create both these positive and negative interactions between microbes within a community. This is a recent study where the authors used phylosphere communities isolated from tomatoes, and they found that they could actually suppress infection by Pseudomonas. So the buffer is without the phylosphere communities present, and this is a representation of the Pseudomonas infection. And as the phylosphere is added either in diluted or undiluted amounts, the Pseudomonas infection decreases. But what they also found is when they add fertilizer to the situation, that suppression goes away. And so there's some sort of resource-mediated interaction here, potentially resource competition. And then on the other side of the spectrum, resources can be used as a positive way for microbes to interact with each other. So in this example, a root endophyte, S. indica, cannot grow in the absence of thiamine, but it can grow when it has this other microbe present. And so that other microbe can kind of act as a substitute for a resource. And so these immune and resource-mediated interactions are all occurring within the context of a plant host. And so it's important to remember not only are these factors affecting the microbe, the community composition, their richness, their interactions, but they're having feedbacks on the plant themselves because these 2 factors are inherently tied to the plant. And then we also see trade-offs in defenses and resources. So these 2 factors can have feedbacks themselves. And when we think about this in a real context, in a plant community or plant population, then we're seeing, sorry for the busyness, but we're seeing these microbial communities repeated all over, and interactions are connecting them. And so 2 factors that can really mediate the interactions within these communities are the plant community and the environmental conditions. And the reason for this is because the immunity or the defenses that the microbes are exposed to depends on which plants are present, as well as their resources, so their tissue nutrient concentration, their ability to gather resources, and then of course environmental conditions can alter all of those factors. And so the research that I'm presenting today thinks about how perturbations to these 2 components of the system affect the microbial communities. And when I'm talking about perturbations, some examples of perturbations for environmental conditions that are linked to human activities could include nitrogen deposition, fertilizer use, flooding, drought, and fire. And some examples of plant community perturbations include species introductions, restoration, succession, farming, and grazing. And so while I'm not talking about all of these, it gives you kind of an idea of where this research sits. So I will be talking about fertilizers as an environmental condition that's being perturbed. And the research that we know about so far tells us a lot about fertilizers potentially as mediating resource availability and competition, less so about this immunity part. So there's the example of the tomato phylosphere that I talked about. One area of research that I'll be building off of is grass viruses and how their interactions are mediated by nitrogen and phosphorus. And then finally, there's been a lot of research on soil microbial community in plant ecosystems where adding fertilizers can change their composition and richness. And then the plant community factor that I'm going to focus on is plant invasion. So I have question marks here because the studies so far, we're not sure what the mechanism necessarily is when a plant invasion is affecting these communities, but we are seeing big shifts in soil microbial community composition when a plant invades an ecosystem. And also we see changes in the prevalence of soil pathogens and mutualists. And so the three systems I'll be using to build on that research include viruses in oat plants and looking at how nutrients mediate those. For the invasion, one system that I'll be talking about is California grasslands and the foliar fungi that are in those. And then third, moving into forest system, I'll talk about understory grasses and their fungal infections. So starting with this system, when one process that really contributes to diversity and composition in microbial and other communities is stable coexistence. And this matters a lot when we're talking about pathogens because of the synergistic effects that we can see from pathogen infection and multiple pathogens infecting a single plant. So in this example, co-infection by pepper mottle virus and cucumber mosaic virus has enhanced symptoms. And this doesn't always happen whenever you have co-infection, but it's a potential reason to be concerned about whether co-infection is stabilized by certain processes. And a way to experimentally test for stable coexistence is by using mutual invasion trials. So in this example, there's two pathogens, and their density is mapped over time, and one pathogen is allowed to reach equilibrium, and the second one is able to invade. So if you have a system where two are able to invade each other, then it stabilizes their interaction because if either one is suppressed down to low densities, they can recover. So the system that I used for looking at nutrient effects on stable coexistence were Barley and cereal yellow dwarf viruses. These are RNA viruses. It's a group that includes several different species. And when they infect grasses, they cause stunting and discoloration and reduce seed production. And they are vectored by aphids. So some of the viruses have the same aphid vectors and some have more specific ones. So what do we know so far about these viruses? One important observation is that co-infection occurs at really high rates. And so that is a kind of reason to motivate it, whether this co-infection is promoted by niche differences or some factor that stabilizes diversity. Another result from multiple experiments is that nitrogen and phosphorus addition in the field affects prevalence. So there's some connection to resources and infection. And then the third finding, which is what I'm going to build off of in my research is that nitrogen mediates co-inoculation of 2 of these viruses in this group. So this figure is showing these results, and one of the viruses, inoculation success, is on the y-axis. And so when it is inoculated under high nitrogen conditions, it doesn't matter whether it's co-infection or single infection. But when it's inoculated with low nitrogen, then co-infection decreases inoculation success. So there might be some sort of competition going on in this situation. So the first approach I took was thinking about that result where co-infection is really common, and I was wondering if these viruses co-occur at larger scales than just a single plant individual, and what factors motivate that co-occurrence. So I started by using a data set that my collaborators collected that included virus infection from 5 different Barley and Sierra yellow dwarf viruses in grasslands in California and Oregon, where nitrogen, phosphorus, and both of those nutrients were added to the field. And I looked at a range of different spatial patterns of these viruses and categorized them from segregated to random to aggregated. So these are just some examples of what these spatial patterns could have looked like. So an aggregation of a single virus would mean that the infected individuals are closer together in space versus more spread out. Virus pairs, so this is the one that I'm going to talk the most about. If they're aggregated, then that means there's co-infected plants, and the plants next to each other have those 2 viruses. And then the third metric I looked at was whether virus communities of plants next to each other were more similar or more different. So this virus pairs result was the one that showed the most interesting because we saw a lot of aggregation between different virus pairs. And this aggregation was motivated by whether or not these viruses shared a vector. So if they have the same vector species, then they're often brought to the same plant or the same local area of plants. And so looking at the scale of this aggregation, we found that viruses like PAV and RPV, the one in the other study, could co-occur at ranges from a single plant to thousands of plants. So then the question is, what is keeping these viruses together? Why aren't they potentially outcompeting each other for resources, for nutrients, like in that lab study? So I followed this up with looking at how nutrients affect co-occurrence at 2 different scales. One is within the host, so co-infection, and then one is at the host population scale. And there might be niche differences operating at either of these scales. In this case, it could be differences in resource requirements, infecting different areas of the plant, and we're thinking about nitrogen and phosphorus as potentially limiting resources for these viruses because they might depend on molecules that need nitrogen and phosphorus. At this scale, there could be certain plants that are good hosts for one virus and plants that are good for hosts for another virus, and then transmission between them kind of keeps the diversity maintained. And coexistence at this single host scale usually automatically feeds to this scale. So if you have co-infection, then you can get diversity at this scale. And then on this end, it's the transmission that can lead to co-occurrence within the host. So I used oat plants in the lab grown in 4 different nutrient treatments, so low nutrients, nitrogen, phosphorus, and an increase in both. And then the vector that I use that works for both of these viruses are pati. And the oat plants had cages on them where we put the aphid vectors, and they were either mock inoculated from healthy plants. They had RPV, PAV, or we had some vectors with PAV and some with RPV, so we could co-infect the plants that way. And we did 2 experiments. In the first one, we had single infection, co-infection, or mock, and then at 8 different time points, we collected virus concentration data using qPCR, a variety of different plant metrics, and then we did transmission trials. So these infection treatments were crossed with the 4 nutrient treatments, and we took aphids, fed them on those plants at each of these times, and then fed them on another set of plants that were all grown in these nutrient treatments. So we could see how the nutrients of the source plant and the receiving plant affected transmission. The second experiment we did, I'm calling the mutual invasibility experiment. So at the beginning, we inoculated plants with one of the 2 viruses. Then we waited 12 days and inoculated them with the opposite virus, or we did a mock inoculation. And this was done across all 4 of those nutrient treatments. And then at 5 different time points, we measured virus concentration with qPCR and the mass of the plants. And so the first result I'll show you is related to the within-host coexistence question. So when we're seeing co-infection in the field, is it maintained by within-host niche differences? And what I'm showing here is the virus concentration from qPCR over time. So the top row is the PAV virus, the bottom row is the RPV virus, and then these are in the 4 different nutrient concentrations. So one thing to note is that the nutrients don't really change the patterns of these virus dynamics very, very much. The ones on the top are either the ones that were singly inoculated, so those are kind of our controls, versus the ones that became invaded. So we call that the resident in the invasion scenario. And another thing you'll notice is that those controls and those resident viruses didn't vary that much. So the invasion didn't knock down the residents necessarily. The third thing is that the viruses were each able to invade each other. And it's kind of hard to see what their quantities are from this scale. So this is showing the virus growth rate of those invading viruses when they're a resident and when they're an invader. And so for both of the viruses, they had higher growth rates into the plant when they are invaders than when they're residents. So that tells us that there are stabilizing factors that allow these viruses to coexist within the plants. So we had some mutual invasion. So then the next question is, if we are thinking about a population of plants, does this scale up? Do we get coexistence at that scale as well? And one factor that could limit that is the transmission. So this is showing the proportion of infected during those transmission trials. So these are the receiving plants, and it mattered whether the viruses were coming from co-infection or single infection. So for RPV, they had the same transmission rate either way. But for PAV, when they were coming from a co-infected plant, it was harder for them to transmit. So even though they're coexisting within a single plant, it's harder to get out. And so that means that potentially at that host population scale, RPV might be doing this, it might be able to invade just fine, but PAV coming into a population already infected with RPV, it might be more difficult. So in order to answer that question, is it hard for PAV to invade, we used a model of a plant population. And we broke up the plant population into healthy hosts, hosts that were infected with PAV, RPV, or co-infected. And then these values along the side are just showing transmission rates. The arrows are subscripts. So they have transmission that is affected by whether they're coming from a healthy host, a singly infected host, or a co-infected host. So we could use the data from our experiments to substitute in for those numbers. And we asked for PAV to invade a population that's fully infected with RPV, so that's the most difficult situation for it to invade. What needs to happen? And this combination of parameters, this Q, which is how hard it is to get out of a co-infected host, and then its transmission rate, had to be higher than the mortality rate of the plant. And we used a value from the literature for our mortality rate, and it's pretty small because the main effect of these viruses on their host is through seed production, not that they necessarily kill plants. And so we looked at these conditions under our 4 different nutrient treatments, and I have potential mortality rates on the x-axis, and then I have that coefficient where it's depicting how hard it is to get out of a co-infected plant. And the white diamonds represent the data that we collected in our experiment. So we've got really low transmission rates and then kind of medium amounts of suppression during transmission. But you can see that all of them are within that co-infected or coexistence space. So if they had landed down here, that means that RPV suppresses them enough to exclude them from the population. And then, of course, this also gives us an idea of how much mortality could we actually withstand in the host population before we start to see exclusion of PAV. So from the results that we also have coexistence at this host population scale. And from this project, we found that PAV and RPV coexist within oat plants regardless of those N and P supply rates that we tested, and that this can scale up to a host population. So the second project that I'll be talking about is with collaborators at Stanford University, and I started working on this when I was first starting my postdoc. And so it's in the context of a plant invasion, which is the second project as well. So I'm going to talk a little bit about enemies and plant invasion. So a hypothesis about how plants can successfully invade is called the enemy release hypothesis, where one important component of that is that when a plant is introduced to a new location, it loses a lot of its enemies, and that gives it a competitive advantage. So that's one dynamic we can see for an invasive species. A second dynamic might be that it accumulates a lot of new enemies in its new range. And that can be called biotic resistance. It could potentially even limit its ability to establish. And then the third process we can think about is that the way that invasive plant can actually influence the pathogens that are within the system. So here we have that invasive plant, the native plant, and the invasive might be a particularly good host for a pathogen, so we might see amplification, and then spillback is when pathogens are then transmitted to the native host. So that's a negative effect that we can see. And then another potential thing that has been observed is suppression of mutualist of the native pathogens. So both the invasive species can be affected by pathogens in its establishment and interactions with the native species, and it can affect the native community. And then another process that invasion is likely to influence is the density of hosts in the population. So one reason that invasives can be a big problem is because they reach really high densities. So if they are increasing the amount of hosts in the area and the pathogen is transmitted by density dependence, then we can see higher pathogen burdens from that. So another way that invasive species can influence. So those studies and experiments that we have seen so far have really looked at the different ways that these connections can occur. But one thing that's missing is how this is changing over longer terms. There's some evidence that the longer an invasive species is in the system, it can accumulate pathogens or it can accumulate mutualists, but we're not sure how these factors are feeding back on the native species and the plant community as a whole over time. And so the question that we asked with this second project is, long after an introduction has occurred, how do native and introduced species influence each other's foliar pathogen communities? And we looked at the types of pathogens that are infecting two groups, the native and introduced group, and then the extent of damage. And our hypothesis was that one of these groups would contribute to spillover, and then we would see overlap between the two groups of plants. and that potentially the receiving host on that end of the spillover could have more damage. So these data were collected from Jasper Ridge Biological Preserve in California, and California grasslands have been invaded with annual grasses for a very long time, and this reserve has both invaded areas and areas with native perennial bunch grasses, and a mixture of both. And so I'm going to talk about data collected from two different experiments. One experiment was more of an observational, where in 2 years, my collaborators set out transects and they collected plants, multiple plant species that I'll talk about in a second, and they measured, they isolated pathogens from lesions, they measured an extent of damage, and then they also surveyed the background plants in these plots. And the primary purpose of this observational experiment was to understand how community composition is affecting pathogen infection. The second experiment was a manipulative experiment to understand how disease mediates competition. And this one was just collected in the second year, so adds a little bit. So in our first year of this one, we had a lot more plants, so we're adding to that second year. And they collected the same data, pathogen ID, damage, and the background plants in the plots. And so our focal species consist of 5 different introduced annual grasses, 2 native perennials, and an introduced perennial. And one thing I'll note is that while we only have 2 native perennials, and in all my results I group these into native and introduced. Even though there's only 2 species there, it's not that one species is disproportionately contributing to our results. It seems like our results are dependent on both of them. And then for our background species, we had the same perennials, but we had a few more annuals in those plots. So generally, the process was to isolate from lesions, and we have about 1 lesion per leaf per plant. So we have one fungal species that's identified for a plant, not necessarily species, but an OTU. And so those were isolated and grouped together. So the way that we analyzed the background community was by using density. The plants in these plots were counted the number of individuals per meter squared. So we have a range of native grass density, which is usually a smaller range than the introduced grass density. And just because of the limits of the number of replicates we have, we grouped them into low density plots, high density of both, high of just native or high of introduced. So the surrounding plants can either be like a mostly native community, a mostly introduced community is how, or both or neither, is how we're analyzing these. And so this is just an example of how I'll show these results. And this is the number of fungal isolates that we have from these different community types. And then the bars are colored by whether they're isolated from a native or an introduced grass. So we do have a lot more natives coming from native communities, but we have a relatively consistent number of our introduced grasses. So our first result that we found is that there was a lot of overlap in the OTUs that were isolated from both native and introduced species. So we're seeing a lot of sharing of the same pathogens. And especially if you get rid of all of these ones, which were only isolated once in our whole data set. And so we looked at if this amount of pathogens that were coming from this generalist group, if that is related to the background community, with the idea that if you're surrounded by natives and you're getting a lot of those pathogens that are coming from natives, then those could be responsible for promoting the transmission. But we didn't see a really strong effect of the background community. In this analysis, or in a community analysis of the fungal communities. So we didn't see an effect of the background community, but we did see an effect of the origin where it was isolated from. So even though there's overlap in which fungal isolates are found on both of those groups, there's differences in the richness and evenness and how frequently they're found. And then some of these were identified, and from the ones that I'm showing here, and a lot of ones in the data set are frequently found generalist agricultural pathogens. So that's interesting. And then we looked at how much damage they're causing to the plants in terms of how the percentage of leaf area that's damaged and also the proportion of leaves on a plant that are damaged. So when we look at percentage of leaf area that's damaged, we saw that when the introduced plants were in these communities that had high density of both species or both groups, they had the highest amount of damage. And so if we just break this down and instead of looking at kind of these categories and just look at the background grass density, that accounts for a lot of this increase. So essentially, when introduced species are in communities that have a lot of individuals, they're getting more infected. And because these introduced species are annual grasses and they're more likely to occur in high densities, we can think of this as sort of a feedback of the introduced species on itself. If we look at the other metric of damage, percentage of leaves on an individual plant that are damaged, then we see a lot higher on the introduced grasses. I'm sorry, the native grasses. So what we have from that study so far is that native and introduced grasses share a lot of fungal pathogens, but there's no evidence of a disproportional influence of one group really promoting transmission. And then the native species had a higher magnitude of infection when we looked at percentage of leaves, but there's potential negative feedbacks where introduced species are really increasing their damage when they get to higher densities. So the third project that I'm going to talk about also goes along with these ideas of invasive species and contributing to the fungal pathogen communities. And this one, we took a little bit more of a manipulative approach. So again, we're thinking about long-term dynamics here. Our experiments take place we did one this year and we'll do one more next year, but we're using mathematical modeling to try to project these to longer-term outcomes. So the invasive species that we're focusing on is called Japanese stiltgrass or Microstegium vimineum. The gray areas of this figure are showing the invaded range, and the darker they are, the longer the grass has been in those areas. So it's in the eastern half of the United States, and this study by my collaborators identified some of the infection on these. So I should have said, sorry, in this image, you can see the foliar leaf damage, and it's caused by this group of Bipolaris pathogens. So the purple one, which occurs a lot, is something that my collaborators called Bipolaris megaspora because it has really large spores. But there's also several other Bipolaris species, that have been found on these. So they called it megaspora here, but eventually it's going to be called Bipolaris gigantea, and it has been identified in the past, but not in the Bipolaris group. And so this is a closer picture of what the lesions look like. It's pretty easy to work with in the field in terms of figuring out which populations are infected because they have such characteristic infection. And when Microstegium dies, it creates this really dense mat of litter on the forest floor, so you can actually go and pick up the litter and see if the leaves were infected the previous year. So with that, we were able to identify some populations for our study that we knew were infected. And then some previous work with this pathogen has shown that it can infect some native species, And so the Gigantea can, and then Bipolaris isolates from this invasive species, Microstegium, that weren't identified as species can also affect native grasses that overlap with Microstegium. Another interesting effect of this pathogen is that it reduces seed production of our invasive species. So this is two different experiments that are very similar, where seed heads were measured at 3 different sites, and fungicide was applied to half of the plots in the site. And you can see when the fungicide is applied, we get increases in seed production. So that seems promising that this invasive species could be suppressed by fungal infection, but the invasive species produces a lot of seeds, so we're not sure if this actually matters in the long run. One of our main questions is, what are the long-term outcomes of this Bipolaris infection on Microstegium and the plants that it co-occurs with? So the beginning half of this is showing what you typically think about in an invasion, where the invasive species is increasing its spatial distribution or population size over time, and the native species is decreasing. But there's generally three different outcomes that can come with infection, which we're assuming accumulates by this time point. So one is that the pathogen accumulates and the invasive species declines, and then potentially, so that's this line, potentially the native species could recover. Another is that there's no effect of the pathogen on the population dynamics. And then the third is one that I talked about earlier, where the invasive species really amplifies the pathogen and it spills back onto the native species, and we get an even more negative effect on the native community. So this first outcome, this paid pathogen accumulation, invasion, decline, what we're interested in knowing is that seed suppression strong enough, and what does it take for the native species to recover? So that would be kind of an ideal scenario. So in order to start to get at that, we're working with plant population models where we can use published experiments, new experiments, and some estimated values to look at population dynamics of Microstegium, the invasive species, and then one native species that we're really focusing on, which is Elymus virginicus. So this is Virginia wild rye, and it co-occurs with Microstegium. And then these are some assumptions that I have in these model results that I'm about to show, and we're hoping to kind of explore more of this model and test some of these assumptions. So one is that in this scenario, only Microstegium is negatively infected, and it's affected in the way that those experiments showed with the seed suppression. Both are acting like annuals, and then we assume some time frames for infection to occur, and then Microstegium we assume is a better competitor, which we've seen that in experiments. So in the scenario where there's no disease, the parameters are set so that Microstegium reaches its carrying capacity and Elymus is essentially excluded from the system. But if we add disease, so this is the time for accumulation, and then there's a pretty quick effect on the population growth rate because of that seed suppression. Microstegium is suppressed, but still under these parameter conditions, Elymus still can't recover. However, if we change one parameter of the model, the carrying capacity of Elymus, we can see that as it gets higher, then Elymus is able to recover, and there's even a scenario where Microstegium doesn't essentially invade at all. And the carrying capacity of Elymus in this scenario is equal to this one, so it's just the disease essentially that's suppressing the invasion. So what we're getting from this is that it is possible to see that paid scenario in our system, but we really need to understand competitive dynamics, both intraspecific and interspecific. So need to know competition coefficients. The second thing that we need to know in order to understand these potential outcomes is how transmission to natives occurs, the rates of that, and the impacts on the native species. So in order to fill in some of those gaps, we are doing field experiments and greenhouse experiments. The field experiments take place in Indiana, so more central part of the Microstegium invasion where we know there is disease. And then the greenhouse experiments are at University of Florida. So in the field, we're asking, will infection lead to one of those three outcomes? Or under what conditions do we see those three outcomes? And then in the reverse, how does the pathogen community change when a plant community is invaded with Microstegium? And then in the greenhouse, we're wondering what are the effects of Microstegium litter? So like I said, it creates this really dense matted litter. What are the effects of litter on Microstegium, Elymus, and their interactions? So my experiments are really focused on this Elymus as a native, but I do want to say that my collaborators are looking into infection on some of the other native species. So in the field, we're working in southeastern Indiana at Big Oaks National Wildlife Refuge. This is a fenced-in area where we have areas that are known Microstegium invaded, and the pink circles all have infection, and the blue triangles are areas where we identified no infection in the previous year and this year. And so I'm going to talk about an experiment we did at the pink circles, which we called the density experiment. And we had 20 plots at each of those 4 different sites. And in the plots, we used PVC grids to lay out different combinations of seedlings that were grown in the greenhouse at Indiana University. So we have, in every plot, we have 3 Microstegium seedlings, 3 Elymus seedlings, and an Elymus adult that we got from a nearby site. And the reason that we include Elymus as an adult is because it's a perennial, and so its competitive tolerance might be better as a perennial than it is as seedlings. And we had plots with just those 7, but we also had plots with different densities of Microstegium, Elymus seedlings, or Elymus adults in order to estimate those competition coefficients. So by increasing these, we can see the competitive effect of each species on each other. And they're proportional to observed densities. So the high density of Microstegium included 64 individuals, whereas the Elymus adult included 8 individuals. And then we crossed all of those treatments with a fungicide application or a water control in order to suppress the foliar fungal disease. So over the course of the summer, we took measurements on those 7 plants to understand plant success, so stem number, height, basal area, and then we did seed numbers on large portions. So here we have little white bags. So we did white bags on the Microstegium to get an estimate of per plot seed production, and we sampled most of the Elymus. And then for infection, we did some coarser measurements, like the number of stems with infection and leaves with infection, and then we scanned leaves for surface area that's infected. And then we're also using microscopy and sequencing to identify the pathogens, or my collaborators are doing that work. So what they've found so far in terms of which pathogens are present, the Gigantea species that they had found in that other study is present on most of the Microstegium and a lot of the Elymus. There's other fungal species that haven't been identified yet on the Elymus. And then the Bipolaris is frequently found also with what's probably a Pericularia species. So there's co-infection on our Microstegium. So this is kind of a busy figure, but it's showing all of those 7 individuals in all of our plots over the course of the summer. And what I want you to take from this, we have the proportion of leaves infected. So like I said, it's kind of a coarse estimate of infection, but the adult infection seems to be peaking earlier in the season than a lot of the Microstegium infection. And that's probably related to the phenology of these plants. So the Elymus are earlier season, they have green leaves earlier, they get going a little bit earlier than the Microstegium. And so we might be seeing some time interactions where the pathogens on these are increasing earlier, and then the Bipolaris is increasing a little bit later. And we didn't see what looks like Bipolaris, on the Elymus until a little bit later in the season. So it seems like there's sort of a sequence of infection that we're seeing. A second result from some earlier season data is that the density of Elymus or Microstegium positively predicts the proportion infected in the plots. So we are starting to relate the plant community to how infected they're getting. So from the greenhouse, like I said, we were asking this question of the Microstegium litter effects. And a couple things about the litter. One is that we suspect that it might be where the Bipolaris is overwintering, so that in the spring, if there's an area with a lot of litter, then infection can start from there. We're not totally sure if that's the process. And then the other thing about the litter is that a lot of Microstegium seed is actually hidden in the stems, and so there can be really large seed buildup in areas with a lot of litter. So we harvested litter from Indiana. This top picture is kind of a dense mat of litter over a log, and we brought it to the greenhouse, and we did 3 different litter density treatments in our pots. We also had a no litter control, which I forgot to take a picture of, so that's substituting. And in each of these plots, we had Elymus, Microstegium, or both of them together. And then we had also an additional treatment where we added shade to see if that affected the relationship between litter and the plants. So this experiment was led by an REU student, Lily Benitez, and she measured plant success in terms of number of individuals, and then we just recently finished up some biomass measurements. And then infection we looked at in terms of individuals infected and identification of pathogens, which we don't have the data on yet. But in terms of infection, in this experiment, it seems like what we observed is not from the litter, because we saw it in our no litter controls. So the infection that we observed is essentially from seeds or soil or some other source that we're not sure. But we still have some interesting results. So one is that as you increase the density of Microstegium litter, you get a lower germination rate from the seeds that we put in of Microstegium. So we have an idea that competition could come in that way, which is interesting. The second outcome that we saw that is that, not too surprising, but Elymus counted in terms of stems, and we'll soon check this with our biomass, does better alone than with Microstegium. So that's something we expected from previous experiments. And then the third one, so this infection I said that we observed, that occurred across all of our treatments, it occurred more frequently when there was live Microstegium present than in the absence of Microstegium. And so this isn't transmission, it's not being transmitted from Microstegium because we didn't, at least not that we are aware of, because we didn't really see any infection on Microstegium. What we think is going on is that this plant has broad leaves, it grows really big and really bushy, and it might be altering the microclimate conditions, making infection more easily occur on the Elymus. And so from this experiment, what we're seeing so far is the Bipolaris gigantea that we saw in other experiments infected our study species, and we're interested in exploring this effect of phenology of the plants in relation to the pathogen community. And then in the greenhouse, we're also interested in pursuing this idea of microclimate changes rather than just transmission as a way that the invasive species could affect infection. So one other add-on to this Microstegium project is one way that we are trying to expand it to more species. So we're just focusing on 2 species of pairwise interaction in all of those experiments, but there are other invasive species, like these shown here, kudzu, garlic mustard, that can become infected with pathogens in their invaded range. And we're working with citizen scientists and land managers to try to collect some more data on this infection on invasive species. So our collaborators are this group out of University of Georgia, the Center for Invasive Species and Ecosystem Health. And they run a website and a suite of phone apps that are called EdMaps. And so these allow people to upload observations of invasive plants, insects, wildlife, and pathogens. And they include all sorts of data like where they saw them, some of the habitat conditions. And so we worked with the people at EdMaps to add another question to their plant surveys where people can say whether or not they saw what looks like damage on them from herbivores or pathogens, and then to take a picture of the leaf with a blue or white background. So that's something that we're trying to get off the ground. If you're interested in collecting data when you're out and about, then this is a fun way to do it. And hopefully we'll be able to get some broad scale patterns of infection on invasive species in their ranges and maybe what habitat variables might be related to that. So with that, I'd be happy to take any questions.